I'm Erica Berg, Director and Senior Editor for Custom Publishing at Science, and I'll be the moderator for this discussion. Our bodies are in a constant balancing act known as homeostasis. The ringleader behind all this? Autophagy, an essential lysosome-mediated degradation pathway that manages cellular viability and death in response to cellular stress. Sometimes autophagy can go wrong, triggering disease. Today, we'll be talking about how post-translational modifications are involved in the regulation of autophagy and what this means with respect to treatments that target diseases. Before we get started, I wanted to give a few instructions about the viewing console. Just to the right of the video window is the Presenters tab, where you can find photographs and biographies of today's speakers. And over there, you can also see, you can log into your social accounts during the webinar to post updates about the event. And on X, you can add the hashtag #ScienceWebinar. After the speaker presentations, we'll have a short Q&A session during which we will answer some of the questions submitted by our live online viewers. So start thinking about some questions now and submit them at any time by clicking the Ask a Question tab also on the right, typing the question into the box, and then clicking Submit. Finally, a thank you to Cell Signaling Technology for sponsoring today's webinar. I'd now like to take the opportunity to introduce our speakers today. I'll give each of them a chance to say hello and introduce themselves. First, I'm pleased to welcome Vojo Deredic. Hello, Vojo. Vojo, would you like to take a chance to introduce yourself? I think you might be on mute.
Yes, of course. Yeah. Hello, everyone. I'm Vojo Deredic. I'm the chair of Molecular Genetics and Microbiology Department here at UNM School of Medicine. And I'm also the director of a center that's called AIM, affectionately called AIM, for autophagy, inflammation, and metabolism. I welcome you all to participate in this webinar.
Thanks so much, Vojo. And next, I'd like to welcome Ruben Shaw. Ruben, want to say hello?
Sure thing. Hi, everybody. My name is Ruben Shaw. I'm a professor at the Salk Institute in La Jolla, California. And I also serve as the director of our NCI-designated cancer center here at the Salk. So I'm looking forward to an exciting webinar today.
Great. Thanks so much, Ruben. And we're going to go ahead and start with your presentation. Please begin at your leisure.
Great. Okay. Welcome, everybody. Well, the focus of my part of today's webinar is going to center around kinases and the role of phosphorylation in autophagy. So to kick us off in the process, what I've got here on the slide is a figure providing an overview of the individual steps that make up the overall process of autophagy. And so many of you will know that this is a highly conserved pathway found all the way through eukaryotes, first worked out in pioneering work, Nobel Prize worthy work in yeast, and then mammalian homologs of all these different components have been cloned over the course of the past 2 decades or so. At this point, the field is mature enough in its understanding of individual steps, starting with stress stimuli that initiate the process of autophagy on the left through a highly conserved kinase complex known as ULK1. It's known as ATG1 in lower eukaryotes. ULK1 in complex with its scaffolding protein FIP200 and adapters ATG13 and ATG101 go on to regulate the PI kinase complex composed of VPS34 as the lipid kinase and Beclin along with associated proteins. This kinase is responsible for the creation of the phosphoinositide 3' pool that is used to catalyze the initiation of autophagosome formation. So the actual nucleating of autophagosomes starts with that complex, which is itself controlled through phosphorylation events that emanate from the ULK complex. Downstream of the VPS34 complex, there are a variety of factors ultimately converging on the conjugation of the LC3 family of proteins. These are homologs of ATG8 in lower eukaryotes. Autophagosomes go on to mature, fuse with the lysosome, and ultimately degrade their cargo proteins. So this is the central biochemical and cell biological processes involved in autophagy. Now, the center focus of my own laboratory is a kinase that was on the right of the last slide. It was all the way on the left, in fact, all the way to the left in the blue box. All the way to the left is a kinase known as AMPK. And this stands for the 5' AMP activated protein kinase. This is a energy sensing kinase. And what it is truly sensing is 5' AMP molecules in the cell, which are, of course, created when ATP is hydrolyzed. So in cells, when energy levels get low, AMP directly binds to one of the regulatory subunits of AMPK, known as the gamma subunits. That induces a conformation change and allows AMPK to be activated. The simplest way to think of this is that this is an energy gauge or a fuel gauge, like in your car. And when energy is low, it becomes activated, and it serves to restore energetic balance. Along the bottom of our slide, we've got images of a variety of different species in which mutations in AMPK itself have been shown to have profound effects on growth and on lipid and glucose utilization and storage. Now, when energy is low within the cell, AMPK is activated and downregulates biosynthetic processes, including protein synthesis, fatty acid synthesis. It also induces catabolic processes, including fatty acid oxidation, but it also modulates autophagy. And for the purposes of today's talk, this is going to be relatively simplistic, but I want to say explicitly for those of you who may be aficionados of autophagy, that it is not appropriate to think of this as really just an off-on switch. AMPK is phosphorylating multiple proteins within the autophagy cascade and is actually modulating up and down different aspects of autophagy. So it is not simply the AMPK turns it on. It is more the AMPK is controlling individual steps, and at some points it may be accelerating aspects of autophagy and shunting and inhibiting other aspects of autophagy is the more global picture. But for today's talk, we're going to talk about decoding individual steps within this pathway. And in brief, my lab has used an approach of combining genetics with affinity phosphoproteomics to try to identify novel substrates of kinases that may be involved in autophagy, as in this example with AMPK. And to make a long story short, we can make mice or cells which are mutated for these kinases and then use antibodies such as the phosphomotif, the AMPK optimal substrate motif antibody, and then immunoprecipitate from lysates with or without a different stimulus, in this case in a mouse even treated with the widely used diabetes drug metformin. You can see in the red arrows at the very bottom that individual proteins are immunoprecipitating with this antibody only in the wild type livers when treated with metformin and not in knockout livers treated with metformin. From that style of analysis, Our lab and subsequently others have found a number of direct substrates of AMPK, including ULK1, this upstream initiating protein kinase in the autophagy cascade. However, it's very important to note that AMPK also directly phosphorylates multiple components of the mammalian target of rapamycin complex, including its critical scaffold raptor. There are 2 phosphorylation sites for AMPK in raptor that are conserved completely across eukaryotes and have now been shown both in plants and in budding yeast to control mTOR activity. Separate from AMPK, mTOR is directly phosphorylating ULK1 on other serine sites, C-terminal in the protein. mTOR is also phosphorylating multiple components of the Becklin VPS34 complex. And so there is truly both a coordination and a coordinated battle between AMPK and mTOR for the control of autophagy within all mammalian and eukaryotic cells. So to make a very long story short, the second major theme that has come out of my laboratory's work in this space is to discover that AMPK is actually not strictly focused around the control of autophagy, but indeed, because mitochondria are the central powerhouses of the cell, and they are responsible for the creation of ATP in most differentiated cells of our bodies, the primary physiological stimuli that activate AMPK are even modest changes or insults to the mitochondria. So all kinds of mitochondrial poisons or shifts and adaptations in cellular metabolism will change mitochondrial output, and that will activate AMPK. AMPK then, in response, directly phosphorylates multiple proteins to restore mitochondrial homeostasis. The first, right in the center of the slide, AMPK directly phosphorylates a protein called MFF, which stands for the mitochondrial fission factor. That results in rapid fragmentation, and I mean within 15 minutes of activation of AMPK, you can get full fragmentation of the mitochondria. That is helpful for the cell to be able to sense and then degrade only those mitochondria which have lost outer membrane potential through a process known as mitophagy. In addition, and in a story I won't talk about today, there is a regulator of the RAG family of small GTPases known as the folliculin FNIP1 complex. And last year, in this year, there's a follow-up paper from others showing that AMPK is directly phosphorylating part of that GAP complex, the FNIP1 protein, which ultimately results in TFEB translocation and activation of the PGC1 alpha coactivator to stimulate mitochondrial biogenesis. So in the end, AMPK is directly phosphorylating 4 different steps of the mitochondrial life cycle, if you will. But for the focus of today's talk, in the last few minutes, I want to shift and talk about AMPK control of Parkin. So many of you will appreciate that Parkin is directly mutated and is the leading genetic cause of early onset Parkinson's disease. Parkin is an E3 ligase, which contains ring domains. It responds to ubiquitin and directly modifies and results in the targeting and destruction of damaged mitochondria. Parkin works hand in hand with another gene mutated in early onset Parkinson's disease known as PINK1. PINK1 is a mitochondrial localized kinase that becomes activated through stabilization when outer mitochondrial membrane potential is decreased. So when delta psi goes down, PINK1 is stabilized. It then directly phosphorylates Parkin, and that is part of the initiating step of mitophagy. PINK1 also phosphorylates ubiquitin on a particular serine, serine 65. And so phosphorylation of Parkin by PINK1 and phosphorylation of ubiquitin by PINK1 were 2 of the first best established markers of the initiation of ubiquitin-dependent mitophagy. Now, as you can see in this beautiful image from Wade Harper's lab from a Nature Reviews Molecular Cell Biology review article from 2018, on the right-hand side of your slide, the process of macroautophagy and the initiation of macroautophagy through ULK1 and the Becklin VPS34 complex that I was just telling you about, that is on the left-hand part of that diagram. And seemingly independently, you've got initiation of mitophagy through PINK1 and Parkin, and through the recruitment of autophagy receptors, selective autophagy receptors such as P62 and NDP, TAXBP1, optineurin. There are a large number of these cofactors, which you'll hear more from Voyo in a minute. If we go on, though, what we surprisingly discovered is that Parkin is, in fact, a direct substrate of AMPK and can independently be regulated by ULK1 under some circumstances. So I won't belabor this, as this was published a couple of years ago now, and the references at the bottom, Hung Lombardo and Science Advances et al. The long story short is that we found a highly conserved serine within Parkin in a region of the protein that had not been well studied, but that lay near some patient mutations. So this serine 108 is this conserved phosphorylation site that we found. It had previously been found in a simple atlas of mouse tissues, interestingly, in brown fat, which is one of the few tissues where mitochondria are naturally uncoupled. While we were doing this work, a beautiful paper from David Commander's lab came out using HDX NMR to provide more information on conformation changes within Parkin. And in that work, they identified a previously unknown region of Parkin required for its full activation, and they named it the ACT, activation element. And you can see that on the left-hand side in light green in the center. And at the end of the paper, Commander's lab notes that this element actually contains multiple previous reported phosphorylation sites. They had never been studied, but dumped in databases like the one that I referred to in that previous cell resource paper. So in fact, the second of those is serine 108, and that is the site that we had found as an AMPK-controlled site. And we went on to do time course studies, as you can see here. And by doing these careful studies, studying each of these phosphorylation events, one can really start to dissect what the order of events are. And so here, using antimyosin and oligomyosin, we can see the induction and the timing of different phosphorylation events. And the ones in green, if you can read that up at the top, those are markers, well-established markers of AMPK activation, AMPK itself, phospho-AMPK, phospho-ACC, phospho-Raptor, phospho-MFF, phospho-ULK1. Then we see in blue known ULK substrates. So Becklin at serine 30 is increased, HEG16L1, another previously mapped ULK1 substrates, also going up within 5 minutes of AO treatment. Notably, further down in red, we see markers of PINK1 activation, such as phosphoubiquitin and phosphoparkin serine 65. Those events are not starting until 30 to 60 minutes after stimulus. So we believe this is a very powerful way for the field to kind of dissect the biochemistry and the timing of how different steps of mitophagy proceed. In the interest of time, I won't go through this data as it's published, but we went on to show that the phosphorylation, the early phosphorylation of Parkin in an AMPK-dependent manner is required for the proper subsequent phosphorylation of Parkin by PINK1. You can also get more information if you fractionate into cytoplasmic and mitochondrial fractions. And all the way up at the top of these Western blots, you can see that Parkin serine 108 is being phosphorylated within 2 minutes in the cytoplasm. There's no parkin at that time that is out at the mitochondria. However, parkin serine 65 in the red is induced at the mitochondria at the 1 hour time point. So we believe these tools will be of great interest to the field. And then I'll just end by saying, as much as the picture may be complicated or feel complicated to some of you, there are yet other targets that ULK1 is phosphorylating. We had used consensus sequence to map sites, and Kunling Guan's lab and other labs have done beautiful work. Sharon Tu's lab has done very beautiful work identifying phosphorylation sites in VPS 15. So I want to draw your attention to this very nice paper from Sharon's lab, identifying what may be one of the most important targets of ULK1 within the Becklin VPS 34 complex. Very nice work from David Rubenstein's lab as well, here in developmental cell in the bottom right-hand corner, showing how ULK1 and AMPK coordinately modulate the PICT5 protein, which is of great interest in a potential key modulator of autophagy. So hopefully all of you like kinases at least somewhat, because you can see that there are a number of them regulating these key events, and that's really the theme of my talk. Another node that I'll say in my last minute here is that there are phosphorylation sites, serines, that you can see in the red that lie in front of canonical LC3 interacting regions, Lear domains. And that is yet another mechanism by which ULK1 or TBK1 modulate dedicated selective autophagy receptors, including proteins like BNIP3, NIX, P62, TAXBP1. So I'll just sum up by saying we know that AMPK is modulating different steps of this. ULK1 is modulating discrete steps, and we believe a thorough accounting of all of these regulatory events will really help lead the whole field towards a better understanding. But I'll stop there and just thank my lab for all the pioneering work, thank our funders and NIH and others, and I'll turn it over to the other half of the puzzle from my excellent colleague, Voyo. Thank you.
Thanks so much, Ruben. And now we'll hear from Voyo. Thanks so much.
Thank you, Ruben and Erica. Greetings to all from the AIM Center. I already described a little bit, a few words about it. It stands for autophagy, inflammation, and metabolism. It has a sort of worldwide reach in terms of collaborators and members. And you can see that on the map. So the game plan for today is to briefly connect with Ruben's talk and give you a less known or less popular PTM as a hidden driver in canonical autophagy. Then the real body of the presentation will be on the principles of membrane ATG ethylation and examples of membrane ATG ethylation. And I'll give you a few data vignettes, basically from studies from our laboratory as well. So speaking of this hidden driver as a PTM, we have shown several years ago that syntaxin 17, sort of the favorite child of many people studying the end of autophagy pathway, fusion of autophagosomes with lysosomes, actually has a more complicated journey through its workings within the autophagy pathway, and that actually through a phosphorylation of serine 202, TBK controls its exit from the ER and participation in the formation of autophagosomes. As an aside, I think cell signaling has made an antibody to this phosphoserine residue, and I think it's available. So now we'll switch over to the membrane ATG ethylation, and this is probably a less known term to the participants in this webinar, so I will have to sort of step back and tell you the reasoning behind this new term that's being introduced, and also to explain that it's not just a term, but actually it's a concept. So we are not changing nomenclature, we are actually adding conceptual layers to what used to be called non-canonical autophagy, which, for a while has been a fig leaf term for many phenomena that did not fit the autophagy paradigm. And here in the center of the slide, you see a simplified autophagy pathway that Ruben has so nicely covered. But going back to these, the accumulating evidence on the existence of these non-canonical phenomena, you have here a citation from Lao Tse that you quote, rather, if you do not change direction, you may end up where you're heading. When I look at it twice on the second glance, it also sounds like something that Yogi Berra could have said. But anyway, that prompted us to actually conceptualize the process of membrane ATG acylation and propose that autophagy is one of its manifestations, but that there are other manifestations. And here you see on the bottom a perspective article in JCB that Michael Lazaro and I wrote together. So this is a little bit of a complex slide, but we'll spend a few minutes on it, basically walking through the workings of the membrane ATG acylation as a concept. So in the panel A and B, you see, particularly in B, that mammalian ATG8s, Ruben has alluded to them already, are related to ubiquitin. Is an overlay, but they do have some special properties that actually make them more prone to modify membranes rather than what ubiquitin does to proteins. So in the panel A, you see that they modify aminophospholipid in lipid bilayers, and that this is basically a bond that's very similar to the isopeptide bond that ubiquitin makes with proteins. In the panel C, you see that there is a membrane ATG acylation and de-ATG acylation cycle, just very similar to what happens during ubiquitination and de-ubiquitination. And this was worked out by Noboru Mizushima. And what you have is really very similar principles to what happens with ubiquitin. And that is that you have E1, E2, and E3 enzymes that eventually put the ATG acylation, or actually ATG acylate the membranes. This is often referred to as lipidation, but I want for a moment to have you suspend that term and think of the opposite, that actually ATG8s modify membranes rather than lipids modifying a protein here. So what you see on a little bit of I think this is a periwinkle blue and some other shades on the E3 ligase. Basically, that's a component that's very important to actually complete the ATG acylation of membranes. A famous example is, of course, ATG16L1, but we have a few more now that I will go into details on another slide. So in the panel E is really the meat of all of this conceptual advance, if you will, of membrane ATG acylation. So, of course, the most famous example is canonical autophagy. And the next ones are sort of, I would call them Magnificent Seven, or you can call them the Dirty Dozen, depending on your point of view. But the first one would be veil or chasm. And this is where proton ATPase actually replaces the VP2 that I think Ruben mentioned as a recognition of PI3P. There's no recognition of PI3P. And actually, these single membranes are being modified by ATG8 proteins, or rather ATG acylated. Mind you, actually, when we say single versus double membrane, there is no such a thing. All ATG acylation, in fact, happens on single membranes, even during canonical autophagy. It's not like two bilayers are being ATG atylated, just one is being ATG atylated. So basically, that kind of cute distinction is, I think, a potential misnomer. So, although well-intended. So all these sort of colorful acronyms that you see listed, they happen in different compartments on different membranes in the cell. For example, in the ER is this EVAC process. Then a little bit older, but very important conceptual advances back then were LAP, LAM, and LANDO that happen in the endosomal and phagosomal system and probably involve NADPH oxidase and such. And then you have a very important concept of membrane repair, that ATG acetylation can do this, for example, on damaged lysosomes and other places. The next one is microautophagy, where, for example, damaged or stressed lysosomes can remodel themselves and remove some things from the surface by creating these intraluminal vesicles. And then finally, a term that was coined a while back, circulatory autophagy, is a catch-all term for a variety of processes that have really been worked out beautifully by Jay Debnath, Sally, and others. And then finally, as a kind of a footnote, but a very important one, is that even lipid droplets can be ATG atylated. And in this case, you don't even have a full bilayer of lipids, you have a lipid hemilayer that is sitting on the bed of neutral lipids. So an important thing that happened this year was the discovery of a new D3 ligase that completely displaces ATG16. So what you see here is a mix of alpha fold and known crystal structures on the panel B. And this is all in this article that actually it's a correspondence in Nature Cell Biology that Dan Klionsky and I wrote that came out earlier this year, that actually sort of advances this idea that there are new E3 ligases for membrane ATG acetylation, basically predicting that there will be more, but actually presenting the known examples. So what you have is VP2 on top, which is already described in detail by many laboratories. I think Ruben touched upon it. And you have the next one is the VATPase that I already mentioned in the context of VALE and the EVAC. And then finally, I think this is the sort of very important advancement in the field, is that TEC-PR1 totally displaces ATG16L1 and recognizes a totally different lipid on stressed membranes, sphingomyelin. And here I'm shamelessly advertising this book that just came out. It has 18 chapters written by some of the best experts in the field on membrane ATG acetylation, and also there is such thing as a protein ATG acetylation. But I will use this while you're looking at the cover of that book, just to give you a simple sort of summary of that you can wrap your mind around it, that membrane ATG acetylation is to membranes to what protein ubiquitination is to proteins. So basically stress damage, remodeling membranes are minded, actually taken care of by the ATG8s and downstream processes that depend on them. And the protein ubiquitination, again, you have a very similar parallel functions in protein management. So now I'll give you vignettes from our laboratory, some of the most recent studies, including those that are unpublished. So here you see Tabata working on this project that started back in 2004, where canonical autophagy was proposed by us to actually control Mycobacterium tuberculosis in vivo. So the arc of this story is very complicated. And there's been a lot of controversy. And I think this is a lovely, difficult, but very important experiment that Tabata and colleagues did. And this is just for a moment to explain the result. If you look at ATG16, you will see that that's that's a protein that participates as an E3 ligase component in canonical autophagy and also in these non-canonical other types of membrane ATG acetylation, such as VAL and EVAC and probably others. So the right side of the molecule, where the WD repeats, to make it simple, is participating in the right side of the processes, whereas the left side participates in canonical autophagy. So if you remove the WD repeats, you will still be able to do canonical autophagy. And we know that because the mouse that stops at E230, the whole body mouse, is perfectly alive, happy, bounces around, and is obviously capable of doing canonical autophagy. However, when you infect these mice with the MTB, what happens is that if you have a full size ATG16, they're protected, but if you have the short size that can E230 that can do canonical autophagy but cannot do these other membrane ATG acetylation processes, the mice crash. And you can see this is a really, in my mind, a lovely demonstration that it's membrane ATG acetylation that actually protects against MTB in vivo. So another story, and this is just a single slide on it, where Jingyu Kasano has done work on looking at the early damage on lysosomes, because we've done some other work with the later time points, and did a mass spec, and she discovered that actually early on there is accumulation of stress granule proteins, and published that in JCB, I guess 22. And the also what is present is GABA-RAP. You can ignore for the moment the other components like ascords that are also engaged in repair of damaged lysosomes. But basically in the right lower corner, you see that membrane ATG acetylation recruits stress granule proteins such as NuFib2 and G3BP1, and then in coordination with galactin A, control mTOR, and other processes that are involved in rejuvenation or replenishment of good lysosomes. So basically, I think the TOR story has been beautifully covered by Ruben already. And here is just, without showing any data, just to bring to your attention this paper by Ruhina Javed, who actually discovered a hidden role for membrane ATG acetylation in canonical autophagy. Most of you will know and that ATG8s are very important for recruitment of selective autophagy receptors so that cargo can be captured, but they're not absolutely necessary for the formation of autophagosomes. What Ruhina has reported, there was a hidden role for membrane ATG acetylation that, just like with the damaged lysosomes here, it maintains autophagosomes in good order so that they can actually progress into fully degradative organelles. So this is the final bit in 2 slides on the membrane ATG acetylation that controls retromer dependent cargo sorting. And I think this is as far afield as one can get from canonical autophagy, where most of the membrane ATG acetylation until recently has been sort of binned into. So what you have here is Masroor and Einar in a thinking pose, pondering over what is retromer doing with the ATG5, because that's the mass spec, a 2D mass spec in which they found many components of interest, but particularly interesting was the retromer. Retromer is composed of VPS26, VPS35, and VPS29, and it sorts many cargoes from the endosomal compartment, often in collaboration with proteins such as SNCSs. Here is SNCS27 that actually sorts glucose transporter. And you can see just the vignette down there in the right corner, where actually the interactions, or rather proximity biotinylation that was by mass spec, that actually pans out when you actually try to see whether they proteins interact with co-IPs. So it turns out that membrane ATG acetylation is required for proper GLUT1 sorting by the retromer, and GLUT1 is a glucose transporter, very important, be it cancer, be it diabetes, be it whatever other metabolic disorder is in question. And this is basically on the left side, you can see that GLUT1 by confocal microscopy really beautifully sits on the plasma membrane. However, when you knock out VPS35, that's the second row, it becomes intracellular, and that's because it cannot make it all the way to the plasma membrane and actually colocalizes with LAM. Totally phenotypically indistinguishable phenotype is if you knock out the bottom row here, ATG5. So it looks very much like the VPS35 knockout. And then if you look at the panels, the quantifications, which are by high content microscopy, the left B and the right B, the left B has all components of the membrane ATG acetylation, ATG3, which is the E2, ATG5 and ATG16 are the E3 components, and ATG7 is obviously an E1, and they are very much similar to the effects of VPS35. However, when you take knockouts into canonical autophagy genes, for example, FIP200, which is a component of what Ruben was describing, ALK1, ALK2 with the ATG13 as well, that those don't matter for GLUT1 sorting. So we've come to the end of my presentation, and after giving you these vignettes and the overall description, I just want to walk you through this in very simple terms, and that is that ATG acetylation is membrane stress and remodeling response. ATG acetylation is to membranes what ubiquitination is to proteins, and I'm showing you that cartoon that was also flashed out by on that slide with the book. And I want to finish on a call to action in terms of, this is potentially where the field may be growing, and that there are many functions of membrane ATG acetylation. I gave you just a few examples, but others have done much more. And these are, I think that there are many of those that are yet to be discovered. So if you're interested, welcome to the club, and I'll end here and turn this over to Erica.
Thanks so much, Voyo. And I'll move on to this slide here. So we are now going to be moving into our Q&A portion of this presentation for our online viewers. There's still time for those watching us live to submit your question by clicking the Ask a Question tab on the right, typing the question into the message box, and then clicking Submit. So let's see what we have over here. So this one doesn't seem to be directed to anyone in particular, so feel free to both chime in. So given our understanding of both pro and anti autophagy implications on disease, such as neurodegeneration, cancer, infection, where do you see the greatest possibility of manipulating autophagy for therapeutic treatment? Ruben, you want to go first?
Sure. I think clearly the data that autophagy function is lost particularly in neurodegenerative disease is overwhelming at this point. So efforts to pharmacologically enhance or restore autophagy, in my opinion, at this time in 2024, seem to have a very wide potential for a wide variety of diseases, but again, particularly neurodegeneration. Separate from that, I do believe that autophagy inhibitors do have a very specific potential in cancer, and in particular cancers, in combination with targeted therapeutics autophagy is a cell survival mechanism in all cells, but cancer cells that are faced with a particular therapeutic will be the cells of the body that are most sensitive to the need of autophagy to survive through the night, if you will. And so short-term use of autophagy inhibitors for cancer, I believe, 10 years from now, will be something that ultimately is clinically approved and will find its use in the treatment of cancer. But for the widest future, I think there are a lot of different diseases that autophagy activators may find a place. What do you think, Voyo?
Oh, I definitely agree with you, Ruben. I think the promise is there. I think once the first breakthrough is made, really through the clinic, it in appropriately sized studies, it will be a happy time for both patients and people who study autophagy. I would just add a little word of caution, and that's not really to derail anything, but I think that we have taken many things for autophagy, whereas they're actually manifestations of membrane ATG acetylation. So I think in some kind of a functional way, it would be very important to differentiate what is the canonical degradative autophagy versus other things that may be just as important, but they're more of a protective function for membranes in the cell and few other things that could be totally developmentally related. For example, I just gave you that glucose transporter. So I think not to diffuse the idea that by focusing on canonical autophagy, things will move forward. I think we just need to keep in mind that not everything that is an LC3 dot is an autophagosome. So, the flux measurements and other things for appropriate drugs first in the preclinical studies need to be properly completed and rule out these other things, or rule in, because they may also participate. So I'll stop at that.
Great. Thank you so much. Next question. Maybe Voyo, you want to try this one out first. What do you feel are the biggest needs in the field in terms of reagents to study autophagy?
Right, that's a great question. I think there is so much. Actually, there's so many good antibodies that a newcomer to the field will have really a blast, rather than like many of us initially going through various steps of figuring out what is a good antibody, what isn't. So that's one great resource, and I don't want to advertise just for cell signaling, but I think they've been, at least in our laboratory, it was always a reliable source. But there are many other companies, and that's one thing. So where are the needs? I think one of the remaining question, and I'm sorry for kind of this sounding like having only one oar in the water, but I think we have these ATG8s, we have 7 in mammals, and we need to figure out what they do specifically. There is a pretty good reason why there is only one ubiquitin, and I'm sure there is a pretty good reason why there's 7 mammalian ATG8s. So, good antibodies, good reagents for that would be really very useful at this point.
Thanks, Voyo. Anything on your wish list, Ruben?
No, I think there are a lot of great tools for phosphorylation, as Voyo was pointing out. I think there will still be new ones that chase down underappreciated aspects. Well, neither Voyo nor I today, like, for example, talked about LRRK2, another very important kinase in the process, and there's other targets still to be known. But I agree completely with his suggestion. I think better tools for that can distinguish between all 7 of the ATG8 family members and can really tease out what tissues and which pathologies different things are coming into play. From that perspective, we are still in the early days of understanding and decoding this field and figuring out at what times is our body using this to try to help preserve and restore function in hundreds of different disease settings. So there's still lots of powerful things. The use of tools that will work for spatial technologies as well as advances in different methodologies come in, the ability to do combined spatial mass spec with spatial transcriptomics, with spatial imaging of all kinds, and having more tools where we can look at ATG8 family members with the phosphorylation site as compared to, for example, target genes induced by TFAB or other modulators of the lysosome. That's one of the things I think is like a future space.
Great,
I'll stop there.
Okay. All right. Another one. With the growing appreciation of non-autophagic function of several of the classical autophagy machinery, do we need to redefine how we measure autophagy? Does anyone want to jump in that one?
I'll let Voyo take that one. That feels right up his street.
Yeah, but Ruben could have answered this just as well, because I think we all sort of, if I may refer to both of us, old timers in autophagy field, and that I look at some of our studies back like 15 years ago, and I reread them, and then I see that potentially we didn't really establish that's the canonical autophagy pathway. And I think in the interest of this moving into the clinic in a proper way, I think we could really not reset the whole field, but at least re-examine some of the information that has been propagated then to the next level, just to make sure that that's actually canonical autophagy. And you know what? There's nothing wrong if actually the effect was associated with these other manifestations of non-canonical, what we call autophagy, but I refer to as membrane ATG ethylation as a concept. So this could be, as they say, when the door closes, it may open a window somewhere else. So I don't think these are dead ends, and we can actually have a lot of fun and productivity by actually looking at it with a little bit more next gen view of what these modifications are. Ruben, if you want to add, I'm happy to.
I totally agree, Voyo. And I think also for people in the field or for newcomers to the field, it's very important to realize that every couple of years, new methods come along, and we discover more of the foundations and the things that we thought were the case based on models, originally, of course, from yeast, and where all the different biochemical steps had not totally been worked out. Once some of those were found in mammalian cells, they made sense, but the picture is more complex than we ever knew. So what we even think of as canonical autophagy is always being, like everything in science, is being updated and modified, and that need not be a bad thing. That doesn't mean that some quote landmark paper from 2006 that is proven completely wrong. It's not that we're proving something completely wrong. It's that we have way more information now, and we can really flesh these things out. And I fully agree with Voyo, especially with ATG8 ylation, that these things were simply not known in the past. And the tools that we had were very primitive. There used to be only two antibodies, LC3 and one particular guinea pig antibody against P62 that the whole field used 15 years ago. So we are in a whole different space now, and there really is truly a lot to learn, and the field will constantly adapt its terminology and how we think of these different parallel processes and pathways. And that's a good thing. That is a sign that the field is capable of doing more detail and truly figuring out what's critical in different cell types and different diseases. I'll stop there.